Biochemistry 401, Lecture 13. We're going to continue our discussion of enzymes. We're going to look at catalysis and the profile of a specific enzyme, carbonic anhydrase, and then we're going to look at enzyme regulations. Enzymes recognize their substrates through complementarity of three-dimensional structures. So, an enzyme and its substrate will fit together. Enzymes bind to their substrates through many weak non-covalent interactions. Generally, these are van der Waals associations in large part, although there might be other electrostatic interactions that are found, such as ionic bonds or hydrogen bonding. And so the binding of an enzyme to its substrate involves the shape of the enzyme and the shape of its substrate. They generally will bind to one another with a high degree of complementarity that brings these surfaces together in close approximation so that you can have optimal non-covalent interactions that are taking place between the enzyme and its substrate. You remember that van der Waals associations are very weak, so the substrate and its enzyme have to have surfaces that follow one another in a complementary fashion, follow each other very closely, for those van der Waals associations to be of maximal strength. So there are two main schools of thought regarding complementarity of enzymes with their substrates. So this is the ability of an enzyme to have a great degree of specificity for its substrate based on complementarity of structure. First of all, there's a lock and key paradigm in which this complementarity of structure exists even before the enzyme and its substrate bind one another. So this is like a key fitting into a lock. This complementarity exists before these guys get together. The substrate just goes in there and binds. The induced fit model says that there might be a little bit of complementarity there, enough to get things started, and then as the substrate begins to bind, there is a three-dimensional structural change in the enzyme itself and in the substrate that sort of mold both of those entities together so that they can form a complementary structure. And so the high degree of affinity arises as the enzyme binds its substrate. The enzyme and the substrate change their conformation to increase the complementarity. These are some characteristics of the enzyme active site. It's a three-dimensional cleft or crevice within the enzyme. It provides an optimal microenvironment for the reaction to take place. The enzyme active site is only a small portion of the total enzyme that provides an optimal environment for substrate binding and catalysis. The other parts of an enzyme offer support, regulation, and can provide binding interactions, binding to other proteins, binding to lipids, binding to sugars. And so most of an enzyme is involved in regulation or binding or supporting the active site. So some enzymes require cofactors in order to function. An enzyme with its required cofactor is called a holoenzyme. An enzyme that requires a cofactor but does not have one, the cofactor is not bound, that one is called an apoenzyme. So we have apoenzymes, that's an enzyme without its required cofactor, and we have a holoenzyme, an enzyme with its required cofactor. Which one's going to be functional? The holoenzyme, not the apoenzyme, doesn't have its cofactor. Enzymes that don't require cofactors cannot be called apoenzymes. That term is only used for an enzyme that requires a cofactor but doesn't have one, and this is important to know. Cofactors supply functionalities that are not found in normal amino acids, and so they allow for processes that a protein on its own cannot do. These cofactors can be classified into two main groups: metals that are inorganic molecules, so this includes iron or copper, potassium, magnesium, manganese, molybdenum, nickel, and zinc. And the other classification is coenzymes. These are organic molecules that may or may not have a metal involved. These coenzymes can be broken into two groups. Those that are loosely bound and act more like co-substrates. These sorts of cofactors enter an enzyme, do their job, and then leave. This includes NAD plus or NADH, and NADP plus and NADPH, and also coenzyme A. We're going to see a lot of these co-substrates in this semester. Now, prosthetic groups are tightly bound groups, and we already saw a prosthetic group when we looked at hemoglobin and myoglobin, and this is the heme molecule. Other prosthetic groups include FAD or FADH2, FMN or FMNH2, pyridoxal phosphate, thymine pyrophosphate, cobalamin, tetrahydrofolate, and lipoate. And again, we're going to see these throughout the semester. We're going to talk about some of these in more detail in the next lecture. So you're going to have to know which cofactor fits in which classification, which are coenzymes or metals, which coenzymes are co-substrates, and which are prosthetic groups. Knowing this will help you as you go on further in the semester. So it's a good idea to memorize this now. It's fairly easy to figure out which ones are metals, and the other ones are going to be coenzymes. Of those coenzymes, you only have to worry about NAD+, NADH, NADP+, NADPH, and coenzyme A. If you memorize those as being co-substrates, you know the rest that we're going to see are going to be prosthetic groups, and this should make it easier for you. So how do cofactors help enzyme catalysis? Well, to look at this, we're going to look at an enzyme that works very, very fast. And we've seen the reaction that it catalyzes before when we looked at blood buffering systems. This is the enzyme carbonic anhydrase. The enzyme is shown in ribbon diagram, and the zinc metal is shown as a blue sphere in the active site. This enzyme catalyzes the hydration of carbon dioxide to carbonic acid. This reaction would proceed pretty fast without the enzyme. However, carbonic anhydrase can increase the turnover to 10 to the 6 molecules per second. That's 1,000,000 molecules of carbonic acid per second. That is pretty remarkable. Here we see a model of carbonic anhydrase in surface diagram. You can see that most of the enzyme is shaded in orange. This is the non-catalytic portion of the enzyme. The active site is circled, and it forms a deep cleft in the center of the enzyme. There's a portion of this catalytic site shaded in purple and another that is shaded in pink. The purple represents hydrophilic residues in the active site, and the pink portion represents hydrophobic residues. In the active site, in the hydrophilic region, is found the zinc ion. This zinc ion has 4 coordination points, 3 histidines, and 1 water molecule. The water molecule is actually one of the reactants in this reaction. The role of the zinc ion is to destabilize this water molecule, allowing it to become more acidic and more apt to release a proton. The first portion of this catalysis is the release of that proton by the water molecule to form a potent nucleophile, the hydroxide ion. This hydroxide ion then makes a nucleophilic attack on carbon dioxide. Now the interesting thing is that this carbon dioxide is held in place in the hydrophobic region of the enzyme. These hydrophobic residues help to position the carbon dioxide in the proper place for catalysis. And so the hydroxide ion makes a nucleophilic attack on the carbon dioxide to form a covalent bond. Finally, water comes in and hydrolyzes this carbonic acid from the zinc ion, releasing carbonic acid to start the next round of catalysis. Now, the carbonic acid that is formed in this reaction has a very low pKa, much lower than physiologic pH, and so as soon as it's made, the carbonic acid is deprotonated to form the conjugate base bicarbonate ion. So the thing is, this is an example of acid-base chemistry. This catalysis involves the production of protons, and if these protons are left in the active site, this could impede the forward progression of the reaction and subsequent reactions. And so it's important to get those protons out of the active site. And so this is done by way of a proton shuttle. This shuttle involves several water molecules and a specific histidine in the active site. This histidine, histidine 64, accepts protons. And so these water molecules and histidine and other residues that can accept protons act almost like a bucket brigade to bring protons out of the active site and deposit them into the aqueous solution surrounding the enzyme. And this proton shuttle is a common theme in acid-base catalysis. So now let's switch gears a little bit and talk about thermodynamics. We can't get away from thermodynamics in biochemistry. So first of all, what is the value of delta G when a reaction is at equilibrium? And the second question is a fill in the blank. The value of delta G must be negative or positive in value for a reaction to proceed in the forward direction. I hope you got these right. We're going to use these as we go on further in the lecture. So here we see a graph of energy as a function of reaction progress. So we know that for this reaction to go in the forward direction, the free energy in the reactants must be greater than that in the products for the reaction to have a negative delta G, a negative change in energy. And so delta G must be a negative number for the reaction to proceed in the forward direction. But this graph doesn't give the whole picture. We can go from free energy of the reactants to the free energy of the products, and we can take any sort of path we want to between those two energies, as long as we end up with the free energy of the product being less than that of the reactants. And so we could actually have an increase in energy before we go back down to yield the lower free energy of the product. This is actually what happens, in fact, in many catalytic reactions. There's actually an increase in free energy as the reaction proceeds. Now, why is this? Well, we saw in the last reaction when we looked at the acidification of water, there is a negative charge that developed on that hydroxyl, and the developing charge has to be countered. And this is where the enzyme comes in. We saw that in the active site of carbonic anhydrase, there were residues that helped to stabilize that developing negative charge, and this has a stabilizing effect during this catalysis. Let's look at this a little more closely. And so here we see what that energy profile looks like. We're going from a lower energy of the reactants to a higher energy transition state, and this change in energy is called delta G double dagger, and it represents the change in free energy from the reactants to the transition state. This increase in free energy in going to the transition state represents an energetic barrier for the forward reaction. Some substrates just simply do not have enough energy to get over this barrier and make products. And so the role of the enzyme is to decrease the energy of the transition state to lower the delta G double dagger. And so more reactants can pass this energy barrier and go on to make products. And so from this graph, we can see that while the change in free energy from the reactants to the transition state, represented by delta G double dagger, decreases, the change in free energy from the reactants to the products, represented by delta G, does not change. And therefore, because of the relationship between delta G and the equilibrium constant, shown here, delta G is equal to negative RT times the natural log of KEQ. We can see that since delta G does not change when an enzyme is added to a reaction, we know also that KEQ does not change when an enzyme is added. This is an important thing to remember because what we're saying is that the equilibrium constant, KEQ, does not change. The concentration of products to reactants at equilibrium does not change, and this is a very important thing to know. Enzymes do not change the ratio of products to reactants at equilibrium. This is a constant for the reaction under those conditions of pH, solution composition, or temperature or pressure. The addition of a catalyst simply changes the time it takes to get to equilibrium. In this graph, we see the molar concentration of products made as a function of time, and you can see that there's a change in scale on the x-axis. There's seconds on the left portion and hours on the right. The catalyzed reaction reaches equilibrium very fast, within seconds, but the non-catalyzed reaction takes hours. Both reactions do get to equilibrium. It just takes a lot longer for the non-catalyzed reaction to get there. Enzymes do not change delta G for the reaction, and they do not change the molar concentration of substrates to reactants at equilibrium. They just change the time it takes to reach equilibrium. We're going to talk a lot about the regulation of enzymes as we begin studying metabolism. So let's start with some strategies for enzyme regulation to give you a good background. First of all, you can physically change the amount of enzymes that are present in a cell or in an organelle by altering enzyme levels, and this can happen through transcription, making the RNA, through translation, making the protein, or through translocation, which is moving the protein from one place to another. For instance, some enzymes work in the mitochondrion, and so you can regulate the effective activity of a specific enzyme in the mitochondrion by just preventing its translocation. And if you'll recall, translation and relocation are ways in which delta ALA synthase is regulated, and this is the enzyme that catalyzes the rate-limiting step in heme synthesis. Did you remember that? Good for you. Enzyme concentration can also be altered through proteolysis. As you can imagine, this proteolysis is highly regulated. Besides changing the concentration of enzymes, enzymes can also be regulated through the binding of inhibitors or activators. Now, we saw some of this just recently when we looked at competition. This is the binding of an inhibitor to an active site to interfere with substrate binding. In addition, inhibitors and activators combine to sites that are outside of the enzyme active site. This type of regulation is called allostery. These effectors can either be positive or negative. Positive effectors increase the activity of the enzyme, whereas negative effectors decrease the activity of the enzyme. And both competition and allostery are very common themes in enzyme regulation. We'll see a lot of this semester. For some enzymes, different isoforms of the enzyme exist. These are homologs that arose through gene duplication, and throughout evolution, these homologs diverged to adopt different characteristics. Some of these characteristics include differing kinetic parameters, such as higher or lower Km or Vmax, or differences in expression levels in location or time of expression within the organism. And we'll see several cases of isozymes later on in this lecture and throughout this semester. Finally, enzymes can be regulated through changes that include breaking or making covalent bonds. These include reversible covalent modifications and proteolytic changes. These are some of the reversible covalent modifications that occur in biological systems. Each of these functional groups can be added to a biomolecule. You are responsible for knowing about phosphorylation, acetylation, and ubiquitination. Phosphorylation is the addition of a phosphate group to a biomolecule using ATP as a donor molecule. Enzymes that transfer phosphate to a biomolecule are called kinases. And the opposite reaction, the dephosphorylation of biomolecules, occurs through the action of phosphatases that remove phosphate groups from biomolecules. We're going to see both of these types of enzymes quite a bit this semester. In acetylation, acetyl-CoA is the donor group involved in transferring an acetyl group to a biomolecule. And finally, ubiquitination. This is adding ubiquitin molecules to a protein to target it for destruction. We saw this earlier in the semester. Ubiquitination and degradation in the proteasome is a good way to decrease enzyme concentrations in the cell, and this is quite common. Another time that proteolysis is important is during digestion. Several digestive enzymes are synthesized as inactive zymogens and then are activated at the right time and the right place. For instance, pepsinogen in the stomach is activated through proteolysis when pH levels are low to form the active enzyme pepsin. In the small intestine, pancreatic enzymes are activated through proteolysis as well. This is part of a protease cascade that we're going to see later on. So don't memorize this cascade right now. You'll have to know it later on in the semester. You should be able to distinguish between a zymogen and an active enzyme for the enzymes that are listed on the slide. It's pretty straightforward, as zymogens generally either end in ogen or begin with pro. So now let's look at allostery, and in particular, let's look at an enzyme that functions in the breaking down of glucose to gain energy. What we see here is actually a dimer. It's part of a tetrameric enzyme that functions in glycolysis, or the breaking down of glucose. One monomer of this dimer is shown in blue and the other one is shown in gray. And there is one active site per monomer, and this active site is bound by a substrate that's shown in yellow. The allosteric site is found in the dimer interface between these two monomers and is bound by an allosteric effector. Now, this effector could either be ATP, ADP, or AMP, because these nucleotides are important in the regulation of the enzyme. Now, here's the question. We have ATP, ADP, and AMP. Some of these are inhibitors and some of these are activators, and they compete for this allosteric site. So let's say, for instance, that the concentration of ATP in the cell is very high. This ATP can outcompete ADP and AMP for the allosteric site to have an effect on the enzyme. Likewise, ADP and AMP also compete for this allosteric site. So, given what you know about ATP and ADP and AMP, which of these nucleotides would you expect to activate this enzyme, and which would you expect to inhibit it? And why does this make sense for an enzyme that's involved in the breakdown of glucose for energy to be regulated in this way? And this is an important question because we're going to be looking at enzymes that are regulated in a similar fashion throughout this semester. So it's a good idea to understand how energy levels in the cell can affect enzyme function and why it's important. So let's think about this for a minute. Okay, so ATP is going to have an inhibiting effect on this enzyme. If there's a lot of ATP around, you certainly don't want to break down glucose in order to make more. It makes more sense to store that glucose for later when times are tough. On the other hand, if there's a lot of ADP or AMP around, that means that the cell is hydrolyzing ATP for energy, and so there's less ATP around and more of its hydrolysis products, ADP and AMP. And so if ADP or AMP bind to the allosteric site, they're going to activate this enzyme to say, it's time to break down glucose now, because the cell is in need of energy. Makes sense, doesn't it? So we said that some enzymes are regulated in response to changes in energy levels in the cell. Serum glucose levels are another important regulatory signal that is used to regulate enzymes in the body. During fasting conditions, for instance, between meals, serum glucose levels will be lower than normal. And so this is a signal to the body that it's time to release glucose into the bloodstream to raise those levels to normal levels again. And so the organ that generally does this is the liver. The liver does this by making glucose from non-carbohydrate sources and also by breaking down glycogen to release glucose into the bloodstream. And so the signal that glucose levels are low has to be received by the liver somehow. Well, this happens through the action of a hormone that's called glucagon that's released from the pancreas when serum glucose levels are low. So glucagon enters the bloodstream. Glucagon binds to its receptor on the surface of liver cells. These are called hepatocytes. Now, the thing is, Glucagon receptors are something called a G-protein coupled receptor. This means that it's coupled to a trimeric protein called a G-protein. So, glucagon binds to its receptor, the receptor changes its conformation, and this change in conformation is transmitted to the trimeric G protein. This causes the trimeric G protein to become activated such that the alpha subunit breaks away from the inhibitory subunits, beta and gamma. The activated alpha subunit then goes and stimulates targets. One of these targets is adenylate cyclase. So the alpha subunit stimulates adenylate cyclase to form cyclic AMP from ATP. So now let's see what we have. In response to low serum glucose levels, glucagon was released. It's bound to its receptor, it's activated the G protein, and now we have higher intracellular levels of cyclic AMP. Now, cyclic AMP is a small molecule that's a very important effector of enzyme activity. One of the enzymes that cyclic AMP interacts with is protein kinase A. Cyclic AMP binds to regulatory units on protein kinase A and causes a conformational change in that enzyme. This causes the inhibitory complexes to fall off and the activated catalytic portion of protein kinase A to become activated. Protein kinase A is then free to interact with downstream targets. And some of these targets are involved in carbohydrate metabolism. Glycogen breakdown is increased. This allows free glucose to enter the bloodstream. Now, as far as glucose breakdown and synthesis, pKa is also involved in the metabolism of glucose use, and so the breakdown of glucose is decreased, and the synthesis of glucose is increased. Again, this allows the liver to release glucose that's made into the bloodstream. Why do you want to decrease the breakdown of glucose in the liver cell? Well, if you're releasing glucose from glycogen, and if you're making glucose from non-carbohydrate sources, the glucose that you've produced, well, you don't want to break that down. You want to send that out to the bloodstream for tissues that need it. The liver can use other things for fuel, and that's what it does. So, the response to glucagon is a decrease in glucose breakdown, a decrease in glycogen synthesis, and an increase in glucose production from non-carbohydrate sources, and also an increase in glycogen breakdown. And so in this way, the hormone signal that started with glucagon results in the elevation of serum glucose levels. And this is an important pathway to understand. Another important theme in enzyme regulation is the presence of different isoforms of enzymes that have different catalytic characteristics, that are expressed at different times, and that are expressed in different places. And to look at this, let's look at the enzyme lactate dehydrogenase. Now, this is an enzyme that synthesizes lactate from pyruvate. This is a redox reaction in which pyruvate is reduced to lactate. This occurs when there's not sufficient oxygen to completely oxidize pyruvate in the mitochondrion to carbon dioxide. Instead, when there isn't enough oxygen, pyruvate is reduced to make lactate. So in mammals, there are 3 genes for lactate dehydrogenase, A, B, and C. The product of A is an M isozyme that's predominantly found in the muscles. The product of B is the H isozyme that's predominantly found in the heart in adults. The product of the C gene is predominantly found in testes. And so we're going to be concerned with the M and the H isozymes. Lactate dehydrogenase is actually a tetrameric enzyme that forms from different subunits coming together. These can be 4 M subunits or 4 H subunits or combinations of them. This represents an electrophoresis experiment in which lactate dehydrogenase was isolated from different adult tissues to show where in the body different isoforms of this lactate dehydrogenase are found. And as you can see, isoforms are expressed at different levels in different tissues. The thickness of these bands corresponds to an increase in protein found in that band. The H isozyme migrates more slowly than the M isozyme, and so those tetramers that have a greater contribution of the H isozyme are higher on the gel than those that have more M isozyme in the tetramer. Therefore, M4 is found at the bottom of this gel and H4 is found at the top. And so in this gel we can see that there are a lot of differences in the types of tetramers that are found. And so there's differential expression of isozymes that correspond to the needs of the tissue. Cells that must operate without oxygen will often express the M4 tetramer. Sometimes they have to work without oxygen. In contrast, the heart itself receives fully oxygenated blood from the lungs. So the heart is not going to be in anoxic conditions unless you're having a heart attack. So M4 predominates in the muscle, whereas H4 predominates in the heart. Isozymes can also be differentially expressed during development. In this diagram, we also see electrophoresis, but in this case, we're looking at only one tissue type, and this is the rat heart. The difference that we see here is a difference in developmental age. Developmental state is shown on the x-axis. We see fetal expression on the left and adult expression on the right. We can see that the M4 isozyme predominates early on in development, and gradually the H isoform begins to be expressed, until finally at 21 days after birth, in the adult, we see the H4 isoform of the enzyme being found. This increase in the production of the H4 isozyme corresponds with the increasing oxygenation of the heart tissue. And so this shows that enzyme expression can change in development according to the needs of the organism at the time. So different isoforms are found in different tissues and also at different times in development. This is the end of Lecture 13. Have a great day.